So if you just look at that, where is the file? Just remember that this is your exam review module. I'm sure that we are recording, just double checking. Okay, that's good. So your file is under the exam 3 review material, and your recording will appear here. And then there will be, next Tuesday, we will have the special topic material review PowerPoint here as well, and the recording. So we'll have 2 recordings, one on special topic and on this, and 2 PowerPoints. So just to let you know. So that's pretty much it, and we can start with the review. And the first thing that what I'm trying to say is, like, I have written the material down on the PowerPoints extensively. So what you now need to do, that this is the trick. You go to those star marked PowerPoint and try to find which quiz question it correlates to. It will correlate to a quiz question, and then study together those questions. It may not appear those quiz questions exactly how it is. Some wordings could be changed or something like that, but this will give you very solid background. So that will definitely work for you. All right, so I have to just control 2 of them. All right, so the first one is the virions, complete common set of assembly reactions. So just make sure that what is being asked. If you are just being asked what are the steps for the assembly for the non-enveloped, so I see that I clearly mentioned this one, then you see that only the yellow steps are the ones what is needed. If it is asked an enveloped virus, then the acquisition of envelope and maturation, because the proteins needed to be inserted into the membrane, that is required. So just know that what is the difference and what is asked. So read the question very carefully, whether it has an asked is non-enveloped versus enveloped. Very important. All right. So after that, we are asking is that what are the cellular machinery that is required for the assembly. So here you have these straight cellular chaperones, transport system, secretory pathway, nuclear import or export machinery. And then, say for an example, if we just asked is that, say for example, right here, you can see that right here in this picture, it shows an endocytosis. Now just remember that this is internalization of the virion. This is not a part of the assembly. So if it's just like, if I just give a question like which is not a part of the assembly, and along with that, if I just add the endocytosis, receptor-mediated endocytosis, that will not be the assembly. You will find a similar question in the quiz, so correlate those things. All right, so after that, we're skipping a couple of slides. Then we are going into, it's very, very specific, so you will see that. So here we have talked about several different type of proteins, a lot of information, but I'm just filtering out that what is required. So here you have a protein HSC70. Here you have a tri-C. Here you have SV40LT. All of these proteins, remember that all of these cellular proteins are ATPases. They all have ATPases to function them, and all of them are chaperone proteins. So just remember that if we just ask that these are all extremely versatile, and if we just ask that which class of function is common to all or exhibited by all the proteins, like this HSC70, tri-C, and ABCE1, so all of these things, if you just be asked all these questions, just remember that your answer will be that they are all ATPase and chaperone function. Very straightforward. All right, so after that, we are looking at that if you just have to look at the poliovirus, first of all, it is a sequential assembly. That's the important part. So just remember that it's a sequential assembly. And if it's asked that what is exactly required, or what is required to make a virion infectious, just remember that this is the important part, virion infectious. Remember that this is required. So this is your answer, VP0 cleavage to VP4 plus VP2. This is required to make the infectious virus particle. Here the provirion is made. This is non-infectious. This maturation to VP0 to VP4, this is a protease cleavage, make the virion infectious. Very straightforward, and you will always be able to correlate a quiz question with that. So study together, then you got the gold mine. That's what it is. All right, so after that, we are looking at the process. What are the different processes involved in the sequential assembly. So just know that a protein scaffold is first formed, and this protein scaffold is degraded by a protease, and then that's basically the overview of that. So the three things right here, it's a sequential assembly. Sequentially, things are added, then the internal protein scaffolding is assembled, and degradation of this internal assembly makes space for the DNA to be packaged. So exactly how it says in these highlighted material, that will do it. So you can easily see that very specific questions or very specific slides, and the review is very specific. Here is the genome packaging. So here, just remember that when we are talking about the genome packaging, remember that the genome is your nucleic acid. So that is being packaged, and the packaging signal is on the viral genomes. And these these sequences, the signals, packaging signals, which are on the genome, which is DNA or RNA, interact with the viral proteins during the viral assembly. So we can just use this sentence as a fill in the blanks. So just remember that your packaging signals is always on the viral genomes and interact with the viral protein during the assembly. So basically, think about that. This is retrovirus HIV. The packaging signals are on these RNAs. Those will interact with the proteins and will be packaged inside. So this is just a similar slide. This is just nothing, same question. It's like I'm not going to ask any of these features in here. But just the idea is that the packaging signal near the left of the inverted repeat and the origin. So you can just see that this is the adenovirus or the SV40. This is their genome, basically. And the packaging signals are on the genome, and see that it is recognized by a viral protein. So that's 4A2 is not important, but these two words, that where the packaging signals are for the genomes, and they are recognized by the viral proteins during the assembly. So that, the bottom line, is the most important part. Just remember that. All right, so fairly easy. You can see that I am not going into the detail of in-between lines. There will be some, but we will see that. Say, for an example, this is a little bit more detailed question that you can see. This is packaging signals of the RNA genome, so the retrovirus, and this is your HIV. So this is the part that I added, which was not on your original slide, RNA genomes, but we're talking about here example of HIV. So these are the 4 features that you have in here. I just extensively mentioned these. You have a size sequence right here near the 5' terminus. Remember that this is the 5' terminus. This is not the end of the genome. This is the 5' terminus of the thing. So you can see that this is the 5' end of the genome. This is not the complete genome. And the genome is diploid. You have 2 copies. Remember that 2 copies of genomes are packaged. And the cysteines and the histidines is box 1, 2 of the nucleocapsid protein. So here, this is the cysteine C and H histidine. They are in the box 1 and 2. These are the interacting. These are the interacting proteins, nucleocapsid proteins. And then there is a basic plus charge amino acid near the N-terminal and in between box 1 and 2 of the nucleocapsid protein. So here you can see that nucleocapsid. So right here, you have plus charge amino acid between box 1 and 2, and between box 1 and 2, and right here. So very specific. And all of these are mentioned signals are needed to be present together for the packaging. So pretty much tells you all the answer. These are the 3 things, and then all of the other are correct. And I'll be like, how much more can I tell that how specific it is? Yeah, I just literally told you the question. So you can see that it like, if you pay attention, you should not regret at all. So if they are this specific, I'm just going to put it around there. So what you need to do is like look at the material over and over so that you remember these specific slides, which are a little more information rich or information too much information in there. All right, so right here, we are good right here. Not asking anything from here. So you can see that we skipped all the way to the vaccinia virus right here. All right, so right here, we can see that there is a signature structure that is formed from the endoplasmic reticulum while the vaccinia virus actually assembles. And this is a crescent. This is the word that is important. This is a signature structure, crescent, which encircles the viral factory or the viroplasm to make the immature virion. So this crescent, this word is the important one. And you may be asked as that which of the following structure is formed by the vaccinia virus from the remodeling of the endoplasmic reticulum. So this is the crescent. And we can just see that the next one, the next slide, I have not mentioned that, but you see that there are filopodia, actin tails, and many other distractors. These could be the distractors, but just remember that these are not for the assembly. This crescent is responsible for the assembly of the virion. This is the signature structure. Actin or the filopodia are not produced from the endoplasmic reticulum, nor responsible for encircling or making the virion. All right. So after that, we are looking at one of the questions. So this is, see that the topic is non-lytic release of non-enveloped viruses. So the viruses are not enveloped and non-lytic. That means that they are budding out of the cells like exosomes or through vesicles. And in this cases, you can see that right here, these exosomes are envelopes around the non-enveloped virus, and this is not considered as an envelope. But the question is that we can just have asked is, or just think about this, exosome formation is not a process for the lytic release of non-enveloped virus. So again, this will be non-enveloped. But as soon as we change the words from non-lytic to lytic, this is not the way it is. See, because what happens, lytic release means lysing the cells. There is no exosome formation or budding. Here, the cell is not lysed. Cell is alive, and you are just budding this process or budding this multivesicular body into the exosomes. If we just ask non-enveloped virus, which is a lytic virus, this means this whole cell will burst and lyse, and the virions will be released. Exosome is not a method for that. So just distinguish these two words, lytic versus non-lytic, what is being asked. All right, so that's fairly quick. So let's see where you go. And here there will be some of the questions which are a little bit more in detail. So, but again, when we're just being asked, I'm just telling you that exactly what you need to know. So for the whole thing, just remember that we can just ask that which of the following pathway is responsible or governed by the PI3K AKT. So you can see right here, PI3K AKT mTOR signaling. If we just ask this, so basically we're telling you the main key PI3K AKT mTOR signaling. If we just ask that, pretty much all of the end product that you are seeing are the effect of this. That means that, see that right here, it will do the actin remodeling. That is correct, through the Rho GTPases. That you need to know. So this is the end thing, actin remodeling. Then apoptosis. This is one of the things that it can block the apoptosis. Sometimes remember that sometimes it can enhance the apoptosis. But again, it can signal. Remember that I am not asking whether it will activate or inhibit apoptosis. I'm just saying that it is whether it is what it signals, basically. So it could be inactivating signal, it could be activating signal, but it will involve apoptosis. And then another thing is autophagy right here, and cell growth proliferation. So we are just asking, see that out of all the complicated one, this is PI3K AKT mTOR pathway. We're just asking the end results of it. It modulates actin remodeling, apoptosis, autophagy, cell growth proliferation. That's all you need to know. So I think that's very doable. All right, so after that, we are asking several questions from here. Oops, where did I go? Okay, so see that I have divided this slide in two different slides. because we're going to ask multiple questions from these slides. So you can just do not have to go many places. So out of this, slide 1 of 2, I just duplicated this slide. So the first question we are asking right here is that the adenovirus modulates the PI3K right here, phosphatidylinositol kinase, and right here PI3K. And what it does, that the PI3K basically does this actin remodeling. And this actin remodeling basically loosen up underneath the plasma membrane so that endocytosis can occur. So we are asking for the mechanism is, say for an example, we can just ask which of the following events or signaling and downstream effects happen upon receptor binding of, So, say for an example, so we can just ask that adenovirus influenza virus modulate the PI3 signaling to gain entry into the cell, and which of the following mechanism involved in the process. So you can see that the cascades through the PI3K remodel the actin under the plasma membrane, promote the endocytosis. So that's the question. So that's basically just shown exactly how it is, so very specific. Then, same slide, second question. So right here is signaling via P3K, and here we can ask specific to influenza virus mediated signaling, what the influenza virus is doing. So first, it activates PI3. PI3, orange and green arrow. I'm just wondering that whether this is, let's see, did I, so I'll see that this may be, so again, orange and, so this looks like red anyway. So this is PI3K, just to understand that. And then clustering of the receptor tyrosine kinase or the RPTK. So upon receptor binding, see that these are the monomers of the receptor tyrosine kinase. And they will cluster on the lipid rafts, and they will also get phosphorylated. See that they are getting phosphorylated. And then actin remodeling from the last slide. This is going to, so I think that I was seeing orange, these arrows, but it is looking like red. So I will just change those basically. And then you have the increased activity of the vacuolar ATPase, VATPase. So the VATPase activity pumps the protons or the H plus into the vesicles. And this signaling is via, see that the signaling is through the RPTK, PI3K, see that both are working in here. So look at the cycle. RPTK, MAP kinase 1,3, that is involved in there. Also PI3K is involved in there. So just remember this, that this is a lot of, so here you have RPTK, PI3K, MAP1,3 signaling. So all of these signaling are responsible for these. And if we just see a word, I mean like this is your hint, and know them all means that probably all the answer choices are given and all of the other or all of them are the answer. So pretty much that's why I'm saying that I'm just like literally telling you the question that what it is. So when you have this kind of know them all means that's what it means. But again, remember that you will be able to connect a quiz question with each of these questions. So look at those questions. So let's go right here. All right, so here also we have asked, is the gene expression in host cells can be modulated by the viruses, except what? And we can just ask is that, say for an example, these are the ones which it does. It does transcription, polyadenylation, splicing, export of the mRNAs. It does the translation, but there is a degradation as well. So if we just ask that which the virus does, except, and if you see the word degradation, you can be a little bit confused, but know that what is being asked is being degraded. That's the important part. So degradation of what is important to answer the question. Remember that the cellular host mRNAs are degraded. That's what the virus wants, not the viral RNAs or viral associated double-stranded RNA. So if we just ask the question, is the gene expression in host cells can be modulated of the virus except, and if you have one of the answer choices, dsRNA degradation, you immediately know that that is not the choice. It degrades the cellular mRNAs, not the double-stranded RNAs or virally associated RNAs. So that's pretty much the understanding of that process. All right, no question from here, right here, no, we are all good. Here you have seen this question that in your quiz as well. Yeah. So you can see that the rationale why the viral infection decreases the protein synthesis initially, followed by a sharp increase in the protein synthesis. So which of the following is the answer? You will see that the first thing that is happening, and each corresponded to the red arrow. Viral infection suppresses the cellular gene expression, cause a drop in the protein synthesis right here. Then you can see inhibition of cellular gene expression inhibition of that coincide with the eclipse period of the virus with no viral mRNA translation. That's why the total mRNA in the cell is low right here. And then followed by the sharp rise in protein synthesis after initial lag period is due to the viral protein synthesis. So if all of these choices are given, you know that all of these are basically correct. So just understand, correspond this experiment. You may not get a picture, and we just can describe this, and you will be able to answer that. All right. So here we have another question on the 5' independent initiation to promote the viral translation. So the question could be that how the viral mRNA without a 5' cap can be recognized to initiate the translation. The answer is by the presence of this internal ribosomal entry site, or the IRES. And see what happens that here, don't confuse by this. See that right here it shows hepatitis A virus IRES requires all, and there is a 5' cap in here. And that is sometimes confusing. Remember that. This is a typical mRNA, cellular mRNA, or viral mRNAs with a cap. And in this case, you need this EIF4E to recognize the cap. But right here, hepatitis virus does not have the cap, but still will require this EIF4E and the intact EIF4G to recognize its iris. That's what it means. So remember that. On the other cases, you do not need the EIF4E or you do need a part of the EIF4G. Here you do not need anything, literally. So these are all recognition of the iris. But here, the hepatitis A virus does not have a 5' cap, but it will need these structures to recognize that. But the overall take-home message is that if the viral RNA doesn't have a 5' cap, iris is the way that it can initiate translation. That's your take-home message. That's it. All right. So right here, we are specifically asking some question that inhibition of the translation upon viral infection, and what happens is the phosphorylation of EIF2 alpha. See that the blue thing everywhere is EIF2, but here is the thing that you need to read. That note the blue EIF2 contain an alpha, beta, and the gamma subunit, and the alpha subunit contains the phosphorylation site. So here EIF2 is referred to as EIF2 alpha only. So in the quiz or question, I will ask EIF2 alpha. It could be spelled out, remember that. It could be ALPHA instead of alpha. So this EIF2, this is EIF2 alpha basically. So what happens that the PKR or the protein kinase R is induced by the interferon when a cell detects the double-stranded RNA. And the activation of PKR by double-stranded RNA leads to the phosphorylation of EIF2 alpha, this one, phosphorylation, and it just blocks the EIF2B, not available, and blocks or inhibit the translation. So the way it can ask is that PKR is an interferon-induced enzyme that is activated by blank. It can be activated by the dsRNA, double-stranded RNA. And then leading to the phosphorylationship of, again, blank and inhibition of translation. As soon as you see that, your second answer will be your eIF2 alpha, activated by double-stranded RNA and leads to the phosphorylationship of eIF2 alpha. So that kind of question can be asked. And you will see that you have a similar quiz question. All right, so the next question that you have with the PKR as well, that what type of viral nucleic acid activates PKR? And remember that it is double-stranded RNA. And see the slide number 82 for additional dsRNA detecting molecules. We're going to see that. But just remember that double-stranded RNA, of course, there is a pact that can activate, but here we are asking what type of nucleic acid. So remember that PKR can be activated by the double-stranded RNA. So we can just ask that which of the following will activate PKR, and we can say dsRNAs, SSR DNA, single-stranded DNA, double-stranded DNA, single-stranded RNA, but you know that it is double-stranded RNA. Single-stranded RNAs will not activate PKR. So that is important. Or say silencing RNA, that kind of thing, that is completely out of the blues. If we have that, those are not the answer. Double-stranded RNA is the answer. All right, so this is the second slide of the same slide. See, that is a duplicate slide. So what is the process of this? Is like a just the whole process, that protein kinase are activation process? Know all of them. That means what you already know. So what are the different things? The binding of the double-stranded RNA, that is one of the things. Dimerization of the two PKR monomers. So here you have the kinase domain, double-stranded RNA binding domain 1 and 2. So this is one piece of your PKR, one piece of PKR, and two of them need to bind next to each other. This is dimerization. Upon dimerization, they will do the cross-phosphorylation. So one monomer phosphorylate the other. So this one will phosphorylate this one. This one will phosphorylate the other one. It is not autophosphorylation. Remember, autophosphorylation will phosphorylate the same, its own. But here it is cross, monomer 1 to monomer 2, to monomer 2 to monomer 1. That's the process. And all of these are basically the activation process of the PKR or the protein kinase R. All right. So let's go right here. Very straightforward question is that which of the following structure develops under the stress to protect the mRNA and inhibited by many viral proteins? That's all. So remember that this is your this portion was not in your original slide. So this is your cellular mRNA pool, and they are just trying to get translated. So what happens upon viral infection? These mRNAs are put into these stress granules. So and there are several other proteins, many proteins they associate with that. And this is basically saving the mRNAs. They are inhibiting translation and saving them for the future. It's a survival strategy. And many of these virus proteins basically blocks the formation of the stress granule so that they can be degraded and the virus can take over. So they are developed under stress, protect the cellular mRNAs. Formation is inhibited, many viral proteins. Straightforward question. No specific names, nothing is required.
All right, folks. So this was the slide that was a mistake in the class. And this is one of the advantage where I post all the videos and I can just make this correction. If it was not, then what I said wrong or showed a slide wrong, that will be wrong forever. Anyway, so what happened here, that you can see that here you are seeing A experiment on your left-hand side, and you see a B experiment. I changed this A experiment to a B experiment and supposed to have these right here, but somehow I just did not save that or something happened and it was right there. So basically I just explained the experiment totally wrong in the class. I would just completely overlook that one. So anyway, so let's go into the detail of that and what we are looking at. So many of the viruses actually increased glycolysis in the virus infected cell. And let's see that what the virus actually is doing. There are many different viruses that can do different things. Some of the viruses increase glycolysis in the infected cell, and some of them may not. So let's take a look. So on the right hand side, this is the original picture from your original slide set. You have a mock that is a control. Here you have an adenovirus, wild type virus here. And here the adenovirus with the E4 gene is deleted, delta E4 deleted gene. So what we are looking at, that these Petri plates contain animal cell culture media. There is phenol red in there, which is a pH indicator. And red color means that they are at a neutral pH or slightly alkaline. Here, the color is being changed, say about in 48 hours, 72 hours into yellow. That means that there is acidic product produced in these Petri plates. And this is the product of glycolysis, which is the lactic acid that is produced in the cell. And this happened in the wild type virus, whereas the control cell without any virus, they just remained in 72 hours. They did not produce that much of lactic acid. They remained red. Now what happens that in the same virus, adenovirus, if you delete the E4 gene, then you can see that the media color remained all throughout red, just like the mock. So what it means is that now the characteristic, if you characterize the E4 gene, it is a positive regulator of glycolysis. This means E4 gene promote glycolysis. As the E4 gene promote glycolysis, more amount of lactic acid, which is the product of glycolysis, is formed. Here, when you delete the E4 gene, you do not see that glycolysis. So this is the case A. Now, this is the question that I asked in one of the quizzes, and this is what it is. Here, what I did, just see that these two things are swapped. In the mock, you have all of them are red. In the wild type virus, suppose this virus is named X in the wild type virus, you see that throughout 72 hours, all of the Petri plates are red. Now, you have one specific gene of the virus, which is, say, a Y gene, and delta Y means that you have deleted this Y gene. If you have this mutated virus and just do the experiment, what you see that after 48 hours, there is an appearance of yellow color in here. So this is different than this. See that this is just swapped. So from here, can you explain the function of the Y gene? Now here, you just have to just understand the rationale is very simple. It is exactly the opposite function of these E4 gene. That will be the exact function of the Y gene. So what it will do when it was present, so Y gene is basically a negative regulator of glycolysis. That means when it was present, glycolysis was not happening. It suppresses glycolysis, reduced lactic acid production. So in this case, it is a decreased glycolysis in the X virus infected cell. When you have deleted this gene, the glycolysis was almost enhanced, as you can easily see. So the question that you can ask here, okay, all right, here you are seeing that some of the yellow colors in here. So here in the mock, which is not infected with the virus, why you don't see that color? So remember that. So what was happening here? There is some virus gene that was under the control of the Y gene that was suppressing the glycolysis. When you take out these Y, that gene is now expressed and it can do the glycolysis. So this is basically the rationale of the experiment, that how you identify the effect of the gene, that you see phenotypic expression of the gene, that what it is doing, and make the proper judgment. So that's basically what it is. So that's pretty much it.
All right, so here is an alternative carbon atom entry from exit from the cycle, and this is very specific. Right here, we're just looking at the HSV, which is taking out oxaloacetate, which converts to aspartate and forms purine and the pyrimidines. So what is the reason, if we just ask that, what is the reason for that? Just remember that the herpes simplex virus is essentially the first thing that it needs to do is to make its large genome. So that means basically DNA synthesis. And to increase the nucleotide synthesis, it needs these pyrimidine and pyrimidine. That's why it taps out this material. All right. So let's see right here. Let's go into the next one. So here from this slide, just remember these basically. So what are the physical barriers? So it's pretty easy because it's not very difficult because you have to learn that in microbiology as well. So mucus, saliva, stomach acid, tear, skin, scab, defensins. Sometimes remember that this is your alpha beta defensins, but just defensins, these are the peptides which makes holes on the membrane. So just remember that. So this is a chemicals in the tears, stomach acid, those are the chemicals. So it's not only physical barrier, but also the chemical barriers. So sometimes we can just ask that which are the physical or the chemical barriers, and which we can ask that which is not of these, and ask some of these. So suppose I just put interferon in here, and that will not be the answer because it is the part of the intrinsic or innate response. So here I say that more clearly. So we just ask that which of the following is a chemical barrier, all of them. And if we just ask that which of the following is not one of them, just remember that saliva, stomach acid, tears, defensins, but mentioned on your slide. So the defensin was not on this slide originally. That's what I mean, that I have added this one, but this is on the previous slide. So that's important. And say, for an example, I said that if we just say that which of the following is not anatomical, physical, or chemical barrier, and if we just give, say, interferon or microRNA or something like that, NK cell, that is not a part of this. So the confusion is that sometimes interferon is in there, and you just try to choose like all of them are, because you think that interferon is a chemical, but just remember that it is not a chemical that is produced upon viral infection. It is not a barrier. So just remember that it is not there to degrade the virus upon or during the entry. All right. So here, divided into next two slides, detail-wise. So first, we are going to look at the RNA detection, the left side. And then the next slide, we are going to look at the DNA detection in the next slide. So here, what happens here that we can just ask is that which of the following will basically activate the PK, or which of the following type of viral nucleic acid will activate PKR, RIG1, TLR3. If you look at that, all of them are double-stranded RNAs. So I very specifically mentioned is like very specifically TLR3, RIG1, PKR, all activated by double-stranded RNA. So very specific question. Cannot be more than that. So that's it. And just look at your quiz question as well. All right. So after that, you have another question. How the viral DNA is detected. The first thing is that DNA is in the cytoplasm, which it should not be. That's the most important part. And upon detection of DNA, a unique dinucleotide is formed by an enzyme, which is known as cGAS. And this is cyclic GMP-AMP synthase, or cGAMP synthase, which is in short called cGAS. And this cGAS produces this unique molecule, which is a 2' and 5'. 2', sorry about that. The 2', 5' cyclic GAMP. So that's what is basically being produced. And remember that 2', 5' cGAMP is a signaling molecule as well. So just remember cGAS. Remember what it produces. It produces a unique molecule, 2', 5' cyclic GAMP. And that's basically the question that we asked for, that how the DNA virus in the cytoplasm is basically detected. All right, so lot of question, and this is the question that I asked wrong. So what does that mean? So before reading the material, just try to understand. I'm just going to explain this one in a second. And so basically what is happening, this is the whole pathway. And the way this works is that, see that the TRAF6 right here, NEMO, I kappa B, they are all added with these ubiquitins. See that? So what they are, TRAF6, NEMO, I kappa B, these are all inhibitory proteins for the signaling cascade, inhibitory proteins. So when they are present, they are inhibiting the whole signaling pathway. So upon viral detection, what is done or the cell needs to do is that ubiquitinate or add these EU. These are the ubiquitins in there. As soon as you add ubiquitin tag to a protein, it is marked for proteasomal degradation. So what's happening that upon viral detection, as the cell adds the ubiquitin to these proteins, they are degraded. That means that pathway will be operating and the NF kappa B will be released out of this I kappa B and it will translocate into the nucleus for the gene expression, and these cytokines are going to be produced. So what happens that what we did that see that all these different protein, they are deubiquitinate. So that means they remove, the cell is trying to put the ubiquitin to operate the pathway, and the viral proteins are removing the ubiquitin. So that was what is wrong in the question. Remember that. So this right here, BPLF1 that we are talking about in this protein right here, BPLF1, which is the Epstein-Barr virus protein, very specific. And what is basically happening is that the question that I has is that so the TRAF6, NEMO, I kappa B, all are inhibitory proteins and ubiquitination, and degradation of the inhibitory proteins are required for the signal transduction to activate the NF kappa B. Viruses block this degradation by ubiquitination. What I had, that one of the question that I have that or the choice that it was like deubiquitination of I kappa B for proteasomal degradation. That was the question, for proteasomal degradation. So again, remember that BPLF1 is deubiquitinating or removing the ubiquitination from all of the protein, of course, TRAF6, NEMO, as well as I kappa B. But what happens that when they remove the ubiquitins, the proteins are going to stay there as inhibitory proteins and the pathway will not operate. But what I did is that I put that right here, just to look at the number 3, I'm just repeating that, deubiquitination of I kappa B for proteasomal degradation. So if you remove the ubiquitin, it's not going to get degraded. It is blocking the proteasomal degradation. So that word was wrong in there, not for. Remember that. Add ubiquitin, degrade. That is what the cell wants upon a viral infection. The BPLF1 is not trying to remove the ubiquitins, which is the cell is trying to do to activate, and that's basically the purpose of that. All of them are is deubiquitination. So just remember that. Spend some time just to understand that. Virus is trying to remove all the EUs so that all these structures or inhibitory proteins stay in place, and this NF kappa B signaling does not happen. So that's the purpose of that. So sometimes why it is confusing, because you're just thinking, why the cell want to degrade NEMO? Why the cell want to degrade TRAF6? Why the cell needs to degrade I kappa B? The reason that they are inhibitory proteins. If they are present, the pathway is not going to operate. So that's the rationale that if the viral protein takes away the ubiquitin, the pathway will stop, basically. So just spend some time and look at the question. I will post this as an announcement as well, and I will just cut-paste the quiz question so that you are not confused. All right, so all good. So here we are just looking at the apoptosis specifically. So we are just looking at that what happens in particularly in apoptosis in very specific cases. So just remember that is formation of apoptotic bodies, which are called these blebs right here, blebs or apoptotic bodies, and the fragmentation of the nucleus. So you can see the nucleus is fragmented right here. And one of the important thing that was in the comment in there is the compaction of chromatin and accumulation around the nuclear membrane. That basically what happens as sharp masses, that's what basically around the membrane, like sharp masses, accumulation around the nucleus, that's happened. And the bleb phagocytosis by macrophage in the tissue. So all these blebs, these are endocytosed by the macrophage. So that's basically what happens. So the only thing that is not shown is the compaction or chromatin accumulation around the nuclear membrane that you just really cannot see very clearly. Well, probably that's what it is. So see right here, this is the chromatin and this is the compaction is happening, and this is all of the compaction. That's probably what the picture basically says. But just know this, these things are happening in apoptosis. All right. So here, this question, and probably this was one of the more difficult question, is that how the ApoBec3 deaminate and what is the process. So let's go to the next slide. So very carefully enlarge your slide and see that what is happening. So remember that the viral RNAs have the Gs, remember that, the Gs and the As. And the reverse transcriptase is first going to make the minus strand, because remember that the virus does have plus strand, a plus strand of RNA. Viral RNA is plus strand. So it's going to make the minus strand, and it's very clear. See that it's 5' to 3', and it's RNA, so you know that it is plus strand RNA. First, it's making the 3' to 5' DNA. So it's a minus strand DNA. And right here, see that from the G, they are Cs, and then you have the CTs. That's fine. Then what happened is the process, which is the deamination. See this, these Cs are changed into Us right here. And when the plus strand is synthesized, they are going to change into, so if you just look at that, the corresponding nucleotides are the A's, and this is basically the hypermutation. So I said that from the fourth para under the notes, you have this. The deaminase activity leads to the formation of deoxyuridine or DEU, most frequently preferred to deoxyuridine or DC sites in the first strand of the viral DNA, first minus strand of the viral DNA. And by the reverse transcriptase, of course. And then the plus strand complement of the deaminated minus strand will contain deoxyadenosine or DA in place of normal deoxyguanosine at such sites. So what happens is that right here, it is only written as a C and the Gs because it's written as the RNAs and the DNAs. And in the quiz question, as I said before, as DNA strand was mentioned in the quiz, the D prefix, which is the DC or DA, that was not there. And we just use the UCA. And if you do not have a D in front of the UCAG, don't worry about that. It will not be a trick question. So it is not needed that it is deoxycytidine or deoxyadenine or deoxyuridine. I have to say that one, but if you just see UCA, you will take that is the nucleotide, and it's not needed. So just remember that. So, but this you need to just understand that what is happening in here. So enlarge that and just correlate and understand this very carefully. Just write it down by yourself. All right. So let's go on here. Okay. All right. So after that, we are looking at NK cell. So right here, remember that the NK cell look at the cells under the scrutiny, and basically what happens, does the cell present, if it is infected, is the MHC class 1 receptor. So here you can see that MHC molecule is with the class 1 receptor. But remember that to trigger killing, you need an additional signaling via separate activating receptor, which can trigger the killing. So right here is the activating receptor. So this is, remember that this is the very important, is that in addition to the viral antigen peptide display by MHC1, which is the MHC1 is displaying this peptide, an activating ligand binding to the activating receptor, which is happening right here, is required by the NK cells to kill the infected cells. So that's your take-home message. So this is your activating ligand, or the activating ligand from the target cell, and this is the activating receptor from the NK cells. This is required in addition to the MHC1, which is showing the viral peptide to the inhibitory receptor of the NK cells. All right. So after that, so very straightforward question from the complement system. Just remember that what is the spice? The variola virus, which is the smallpox virus, produced this smallpox inhibitor of the complement enzyme, contributor of the high mortality, and this is implicated, or this is a protein, or this happens in the complement system. That's the question, is that we can ask that which component of the immune system is inhibited by this protein or the spice. So just remember that your answer will be the complement system is basically inhibited by the spice, which is basically a protein produced by the pox virus. All right, so the next question is, I have just added these part in here. Here what you are looking at, that a dendritic cell is an antigen presenting cell. You already know that, but just remember that the dendritic cell can also get infected. And this is the most important part. I just wrote other viruses like LCMV, not only HIV, can infect them. In that case, it will load the viral peptide, or the LCMV, it will load the viral peptide to the MHC1 and get killed by the CD8 or the cytotoxic T cell, and not MHC2 to activate the CD4 helper cells. So we'll correlate that with the slide 106. So what do we mean by this? So suppose this virus, this dendritic cell, it basically is processing a viral antigen for antigen presenting. When it will antigen present to the CD4 T cells, it will load the viral component on its MHC2. MHC2 presenting to the CD4 T cells to alert the immune system. That is the normal function. But if the dendritic cell by itself get infected, not capture the pathogen, itself get infected, in that case, the cell commits basically suicide. How it does that? Instead of loading the viral peptide display molecule to MHC2, it will load onto the MHC1. And MHC1 can get killed by CD8 cytotoxic T cells, or even it can get killed by the NK cells if it's loaded onto an MHC1. So that's the important part, and we will again revisit this question somewhere else, or slide 106. We'll come to that. So let's see. All right, so right here we are looking at that, let's see. Yeah, so this is basically the continuation of that question. So this is 106, yes. So this is the slide that we corresponding to the previous one. So here, just remember that what is the normal thing for the dendritic cell to do? MHC class 2 expressing antigen presenting cell. If MHC class 2 shows the viral peptide, it will activate a CD4, which is the T helper cell. So CD4 T cells, aka helper cells. This will activate. But when the antigen presenting cell is by itself infected, target cell means that the cell is infected, it's going to show the viral peptide by MHC1. that will bring the CD8 expressing cytotoxic T cell, and this cell will kill this one. Even if it is an immune cell, it can get infected and it will be killed. No matter what, even B cells can be killed if it is infected. So that's the important. Immune cells like dendritic cells can get infected as well. In that case, they will present the viral antigen by MHC class 1 and will be killed by the CD8 plus or the cytotoxic T cells. So that's your question and correlation with the previous slide. All right, so here we are looking at that what is going to happen in the process of this presentation of the antigen in the MHC class 1. So remember that it is produced in all nucleated cell upon infection, all nucleated cells, including the antigen presenting cells, of course, that we just talked. And these antigenic peptides are loaded onto the MHC1 for the display on the cell surface. Say, for an example, this is related to directly the question. Say, for example, in a skin cell, say for a keratinocyte, is infected with a herpes virus like an HCMV, the cell will try to display the viral peptide by MHC class 1, but an antiviral protein US3 in HCMV may block the process. But still, what is important is that we can just describe that process that in this case, what the cell, so I can just describe this whole process. Is US3, HCMV, keratinocyte, you do not have to remember that. I'm going to give those to you. This is was a quiz question as well. If we just ask that this is the infection basically happening to a cell. And what we are asking that if it is infected by a virus, here just basically what it is, no matter what, the cell will try to load the viral antigen onto MHC class 1. So that is what is important. That's what we are looking for the question. So correlate with the question that we have in the quiz and you should be golden. All right, so after that we are looking at secretory antibodies. So here the secretory antibodies are the IgAs. They are very important because they are on mucosal surfaces. It's like saliva lining the nostril, mouth, urinary opening. Wherever there is a moist membrane, they are lined by mucus, and you find this secretory IgAs. And remember that normal Ig or the immunoglobulin is Y-shaped, and see that there are two Ys back to each other. What it means that they are in the dimeric form. This is what is important. And there are some other, most of them monomeric, but this is dimeric. You know, IgM, if you remember, is pentameric, but I'm not asking. Very specific. IgA is the secretory antibody found in the dimeric form on mucosal surfaces for critical antiviral defense. And try to find that what is the secretory antibody and how it is found in that case. So that's it pretty much. All right, so let's see right here how we're doing with the time. We're doing great. So here, basically, what we are asking is the question that the adaptive immunity provides memory. That's the only thing for take-home message. This is a very, very simple question. So basically, we can just ask is that, say, for an example, the immune memory of the adaptive immunity protected the 1918 Spanish flu survivor from the 2000 H1N1 pandemic strain. So if we just ask is that which of the following feature of the immune system contributed towards the immunity of the Spanish flu survivor against the H1N1, your answer will be the memory of basically is the answer for the adaptive immunity. So just remember that it has the immunity. So this is very straightforward question. All right, so let's go into the pathogenesis. All right, so very straightforward questions. You can easily see that just know the definitions, capacity of a pathogen to cause a disease in a host, know the definition of the severity or degree of pathogenesis. Just remember that. Now remember that this was one of the quiz questions and some of them in the first attempts missed those. Viruses can be virulent, avirulent, attenuated I put another one as a mutant, and nobody thought about that. That could be another choice. But remember that mutant virus was not in this list. But you find these mutant viruses, which is different from the wild type virus, is all over the place. And all the attenuated strains are basically a mutant form of the wild type virus. So the viruses, if we just have a question like this, viruses can be found in which of the following form? Virulent, avirulent, attenuated, mutant. Do not choose mutant as that is not found. They will be found in all different forms. So that's basically the answer. All right. So here, what we can ask, and if you remember that we asked a question a little bit different from that what is being asked in here. Basically, the principle of the experiment was asked in a similar quiz question. So what is basically I asked is that in a hypothetical case, a gene knockout can stop production of certain receptor, but in many instances, the scientists have found the viruses can use an alternative receptor. So here you have a very specific example with alpha-2,6 when they knocked this gene, which is basically responsible for making these bonds of the sialic acid. The scientists found that alpha-2,3 basically bonds. But I can just give you a critical thinking question that you have a virus X, and then the scientist thinks that the X receptor is responsible for the viral entry, and then knocked it out. And then they found that even after the knockout, the virus can infect. What will be your conclusion? Your conclusion will be the virus can use an alternative receptor. That's your take-home message. If the knockout doesn't work, an alternative receptor can be used. All right. So here is just a very typical questions. This is just a recap type of question. Reappearance of the cold sore by the herpes simplex virus. This is basically the latency. Very straightforward question. Nothing specific. Is just tell you that what happens, that the cold sore happens, and if we just ask that which of the following best describe the herpes simplex virus infection, we can just give you acute, latent, persistent pathogenic, or persistent asymptomatic, something like this. And just remember that the latency or or latent herpes virus, or the latency is the cause for this. All right. All right, so right here is the varicella zoster infection, and a very specific question. Just remember that what happens is that after the primary acute infection, they go to the sensory ganglia right here. So see that the whole thing is the sensory ganglia, which contains these individual sensory neurons. So again, if you just want to recap from your anatomy, physiology, or any biology or something, ganglia is collection of the cell body. So inside the ganglia, there are hundreds of individual neuron cell bodies. Remember that. Only the cell body is in the ganglia. So it is a kind of like, if you just anatomically look at the piece, you will see that there is a bulged mass in there. So this is just only the cell bodies. And the axon come out of the ganglia. I mean, like these are the unipolar, so they have just only one axon. But if it's a dendrite or axon, they will come out. So this is the ganglia, and the latency in the ganglia, so means that right here, these are the neurons in which the virus will remain and just hide for the rest of your life. And the reactivation can cause shingles. So remember that varicella zoster is basically a herpes virus that it causes chickenpox, but the reactivation causes the shingles. All right. So here also another critical thinking question. You may have seen this one in your quiz as well. So here, what we are looking at, we can just describe an experiment that just how it is. That you are just taking this virus and you have just given 100,000 PFU intraperitoneally, and if you give one PFU intracranial. And then what you do, the important part is that what I'm going to ask here that once the mouse is dead, you did an autopsy to the mouse and look at their neurons in the brain, and you did not find any virus in the neuron. And how can you explain the result? So what are the different things? So first of all, what happens is that we are going all over, and then we're going to go to specifics. So if this is the case, this is the experimental outcome. Virulence of LCMV depends on the route of inoculation. So you can see that intracranial versus so route of inoculation is important. And this one is the important, is the blood-brain barrier can't be breached by LCMV. Why this is? Just remember that. If you give the virus intraperitoneally, it should circulate through the body and it will try to reach the brain, but it does not. If you give it into the body, it is not going into the brain, so it cannot be breached by LCMV. So the blood-brain barrier contain the virus in the body, cannot get in there. So if you just give them intracranially, that means that you have to inject the virus directly to the brain to kill the mice. So that's why you conclude the blood-brain barrier can be breached by LCMV. And the visceral organ tissues are not much susceptible or permissive for LCMV. See that you have given 100,000 PFU, but in the body, why the mouse is surviving. That you can conclude this, the visceral organs or the bodily organs are not much susceptible or permissive for the LCMV. That's why it is surviving. This is also your conclusion. Now, this is what I was talking about. By autopsy, if no virion is found in the brain neurons of a dead mouse, it can be concluded that the glial cells, this is a little bit extra, glial cells, which are the neuron supporting cells, or meningeal fibroblast of the brain of the nervous tissues, are infected, leading to brain inflammation. So most important part is just remember that 60% of your brain mass are non-neuronal cells. Only 40% of your brain is neurons. The rest is other supporting cells, which are glial cells. Glial is basically a generic term. So the glial cells are the supporting cells. And if you remember from that, you do not have to remember, but astrocytes, oligodendrocytes, microglia, these are the different cells which make, give nutrition to the neurons. The astrocytes give nutrition to the neurons. The oligodendrocyte make the myelin sheaths. Then the microglia, they basically are the surveillance cells or the macrophages of the brain. These are the supporting cells. And of course, you will have the meninges or the lining of the brain. You have the dura mater, pia mater, and arachnoid mater. All these meninges will have epithelial cells or fibroblasts in them, and these can get infected, and that can cause brain swelling. Brain has a small space. It cannot go anywhere. That pressure can kill instantly. It's lethal sometimes, brain swelling. So this is basically it is. And the observation can also conclude that the neurons are not susceptible or permissive of LCMV as well. If you don't find any LCMV in the neuron, they are not susceptible. So a detailed experiment, but just remember that a couple of questions could be a little detailed like that. So just take your time, and if I prepare you this way, if you spend time, you should be able to answer these questions. All right, so let's go into toxic viral proteins. So just remember that how the NSP4 basically works. So remember that NSP4 blocks the sodium glucose luminal transporter. If it blocks the sodium luminal glucose transporter, means the sodium is staying in the intestine. And sodium as stays in the intestine, the chloride also going to stay in the intestine to maintain its electroneutrality. More salt in the gut means water is going to come out from the cell. On the other hand, NSP4 activate phospholipase C, which elevates the calcium level. This is a signaling molecule. Remember that calcium is an intracellular signaling molecule, very, very important. And that elevated calcium will basically release chloride, more chloride, into the intestinal space. That means that it will pull water from the cells to cause diarrhea. So more sodium chloride in the intestine, lumen pulls water out of the intestine. So just remember that it blocks the sodium glucose luminal transporter, activates PLC, elevates calcium 2 level, and that calcium 2 level signals chloride to be transported or secreted into the gut or the lumen of the intestine. All right. So here is a very specific question that in the SARS-CoV-2 infection, what is the killer? Remember that cytokine storm is basically by the delayed IFN response. Delayed IFN1 response is basically the cytokine storm leading to the immunopathology. So that is very, very specific. Just remember that. All right. We'll have some questions regarding these, but it will be in the special topics, not from here. All right. So here we have some questions regarding, you will see these questions right here. So basically what happens is that the TLR3, if you have a double negative TLR3, which is basically a homozygous deletion mutant of TLR3, with the impaired TNF alpha production, is more resistant to the West Nile virus. And the question is that why is it so? So just remember that what happens that the TLR3 induced TNF alpha basically compromises the blood-brain barrier, and the brain infection by the WNV proves to be fatal. So what do we mean by that? In the wild type, upon infection, where you have the TLR3, it detects the virus, it is going to produce the TNF alpha. It compromises the blood-brain barrier, so the virus goes into the brain. And how we also know this Evans blue is the dye that was used. And if you have a blood-brain barrier, the dye cannot reach the brain. So here you can see the dye readily can reach the brain. That means the blood-brain barrier is compromised by TNF alpha, which is produced as the consequence of the TLR3 signaling in the wild type. Here in this one, TLR3 is deleted, is homozygous, it's minus, means both the alleles of the pair of chromosome, TLR3 is not there. So this mouse cannot express TLR3. So in that case, there is no TNF alpha production, and it can maintain the blood-brain barrier, you can easily see. So very little even after 5 days. So that's basically the question, that rationale of the experiment. Another experiment is the fatal shock syndrome. Just remember that the pre-existing, the heterotypic antibody binds the virus and conjugates are internalized by these Fc gamma receptors on the monocytes. And this leads to the infection of the monocyte for the high amount of viral production. So this was also, you will find that this was a question that just the mechanism that how this fatal shock syndrome basically happens with the second exposure. What is the cause? This is basically the cause. And I will just probably, we are just almost done. False negative tuberculin TB test is by the immunosuppression by the measles infection. There was an infection. So if a person is not having a TB test positive during the measles, there is no need to think that it is correct because of the immunosuppression as well. I think that's it. Woo, finished it. That's good. All right, so that's it pretty much. So just remember that you will see probably just many of you are thinking it's like, okay, all right, I have seen this question in the quiz. I have seen this question in the quiz. Yes, you have seen this question in the quiz. That is your clue to do well in the that is your clue to do well in the test. So do that, and we will do the honors slides